Welcome to SporeSwaps Podcast, episode 9. I'm here with Dr. Edward Grand. We're going to talk today a little bit about Dr. Ed, his shop, and we're going to talk about selection and breeding and isolations. So, welcome. How you doing, Dr. Ed?
I'm doing pretty good. We're just going to talk a little bit about some small topics. Should take 3 or 4 minutes. Selection, breeding, picking a phenotype, maybe Mon crosses, who knows? Stabilization, senescence. Should take about 5 or 6 minutes.
Yeah, so we're gonna try to cram a lot of talk into an hour, but we figured that some viewers may not know Ed yet, and so I wanted to talk to Ed a little bit about who he is and his history, and kind of like find out a little bit about yourself. But so I know you're in Thailand, but take me back to when you were in America and before you made that move and why you made the move and stuff like that.
Yeah, well, I guess I started, long ago when I was a little boy in Michigan. I grew up in the woods, and so I always, I messed my hair up already. I kind of grew up, fishing in Michigan, hunting and whatnot, and outdoor lifestyle, and I just finally, I kind of found mushrooms out in the woods. When you get bored wandering around the woods, and you finally, when I was a teenager, I started figuring like I could eat some of these things probably. I grew up in Michigan where morel hunting is like really, really like everybody does it. Even 30, 40 years ago, you find some weird morel growing out by the burning barrel or under some apple tree. And, of course, everybody's like, well, you can eat it maybe, but you should figure out and ask somebody, and you've got like what we call beefsteaks, the gyromitras, the false morels. And then so that kind of freaks you out. And you're like, oh, I don't want to just go around eating random mushrooms that I find. And then I think it was in a library or something. I found one of these Alexander Smith books. It was like the Mushroom Hunter's Guide or whatever. I remember it was this tall, weird, like brown covered book, and it had, I don't know, maybe 50 or 60 random mushrooms in it. Some were edible, some weren't, but it was only like 50. So mushrooms, and so like you find mushrooms, and you're just like, I don't. This one's not in the book. So then you get the Audubon Society book. And then if you got, you're getting real fancy, you get like David Aurora's like Mushrooms Demystified, which is, I don't know, at the time about 40 or 50 bucks, and then you just start accumulating books. And then you're finally like, well, I don't know. Remember, this is way before all this internet stuff, right? Before iNaturalist. I'm talking like 35 years ago.
Yeah.
And so you just learn from books, or I found a club, the Michigan Mushroom Hunters Club, which I believe is still like operational or whatever, but I don't know how active they are and doing actual forays and stuff like that. But they do have, I think they have a Facebook group, but it's morphed and kind of been taken over by, I think maybe the more the cultivation side of the hobby. But anyway, yeah, then I was just, doing my normal life thing. I went to school, got a degree that I never used. I had a job that I didn't really like, but I did work in a medical lab for about 3 years, and that got really boring after a while. I was doing all these things that sound fancy, like sequencing and northern blots and ELISA assays and all this shit. But it was like, things that it was like a topic. I was working for a medical doctor, and I don't really like humans that much in general. So I was like, this is really, really cool stuff, but what can I do? Like, what can I do that I really want to do? And that's going to, like kind of get me excited. And that at the time was morphing into mushrooms. And I started even before that. I cultivated oyster mushrooms on straw and cubensis, but it was one of the kind of long stories. There was some close calls that I was like, what? I don't really want to end up in prison. And I don't know, like, it's like that back in the day, things were a lot more iffy than they are now. Yeah. And so I stopped doing that for a little while, and then just, I don't know, I'm jumping all over the place, but otherwise this would be like an hour long. So I ended up going back to school to study mushrooms.
Okay.
But it was the very, very boring aspect of mushrooms, the taxonomy and the nomenclature of Lentinus, which used to contain shiitake mushrooms, but they've since been segregated into a different genus, Lentinula now instead of Lentinus. But there's still some Lentinus that people eat, but that was what I did my work, my PhD in. And then I moved to Thailand after that to try to find new species of mushrooms. I did that for like a year or two, and then realized that also wasn't really, like, the resources aren't really here. Some of the, when you're on the other side of the world, communication is a bit of a problem. And even where I was living, there wasn't really a internet connection, and everything now is through the internet. So, even sending emails and stuff like that was impractical. So I found my girlfriend at the time found an ad in the Bangkok Post for teaching. And so I applied, and like a day later I had a job teaching. So I've spent the last 18 years doing that, teaching chemistry, which was my undergraduate degree, was chemical engineering. So I had like a little bit of chemistry, a little bit of medical stuff, and then the mushroom, kind of like the more intense mushroom stuff. So I've had my kind of foot in the whole like chemistry, psychoactive, like mushroom world for like 30, so maybe more like 35 years. So I've seen all the trends and the kind of new things that people do, like crossing mushroom strains and things like this. And I've seen that going on for, a long, I hate to even like utter the words, but, snake venom and things like this, and things like that. Back in the day, that was like what supposedly people did, people were selling snake venom, shit like that, Yeah.
So when you moved to Thailand, and you said that you went to Thailand to try to discover new mushroom species, like, were you actually moving there to hunt, like try to find new wild psilocybes, or was it to find just species?
No,
not
in
at
general.
Just
I lived. No, the problem is that 20 years ago, and even today, I hate to say it, but that is academic suicide.
I got you.
Like if you go anywhere near psilocybes, unless you're somebody that's well established, or you're like, Gaston Guzman, or you're a big person. Like if you just like study psilocybes in academia, you're not going to get funding. You're not going to get an advisor. You could probably squeeze an academic career out of it these days, but not 20 years ago.
I got you.
You would have immediately got lumped into, it's like your coworkers that don't smoke weed. Like you probably don't tell them you smoke weed. It's like when I would go to mushroom, I've been to mushroom forays, and even places like Telluride, man, 20 years ago, like the magic mushroom, that was in like the back room.
Yeah.
Like, that wasn't like a thing like it is now. So if I would have said to my boss at the time, like, hey, I want to come to Thailand to study magic mushrooms, they would have just been like, no.
Oh yeah. So when I asked that, I meant more like for you personally, like, was that part of why you wanted to do it? Like, you would never divulge that to somebody professionally, you know what I mean? But I didn't, didn't know.
No, that literally had nothing to do with it.
Okay.
No, I'm a mycologist. I've studied different genera. Like, I love all mushrooms.
Yeah, me too.
I to be honest, I found the genus psilocybe a little bit boring at the time. It's not really very diverse.
Yeah.
For somebody who doesn't study mushrooms, like, they don't realize it, but the genus psilocybe has not a whole lot of diversity in it. I know we think because we grow exotics and stuff, but there's other genera. Like one of the students where I lived was studying the genus Marasmius. There's literally like hundreds of species here in maybe not hundreds, but there's much more interesting genera of mushrooms than psilocybe. But again, it's one of those things, like, most people don't care if you can't eat it, if you can't sell it, and it doesn't get you high. Like, people don't care about it. So that's one of the, and as far as that goes, that kind of comes to grant money and stuff also. Like if you're going to be an academic and you're going to buy microscopes and pay for field trips and, you know, hotels and equipment, like you've got to get grant money. So that's a tough sell in the mushroom world, unless you're going to do something that's going to make money. It's like every other business, you know, like the people that are giving you money need to see some ROI, you know, like return on their investment. And in academia, that's basically you publishing papers. Going back to psilocybe, like pretty much it's kind of like a done. It's I don't want to say it's saturated, especially at this point. There wouldn't be you're not going to go out into Thailand or any other place where humans have been and find like a new species of psilocybe. It's not going to happen. Well, there are some caveats to that, but I don't want to get into the nomenclatural and taxonomic issues. But most of the psilocybe species have been described.
Yeah, I mean, I would have to resonate with what you're saying too, because I mean, there's pretty much only one wild psilocybe that grows in this area, and that's Avoidiosus stadiata. And they have a pretty ranging morphology, but generally they look like most exotic psilocybes. You know what I mean? It's not you're talking the more science side, but visually, like there's not very ranging aspects of the way that they look. And I love foraging for every type of mushroom, and there's so many more interesting variants out there that have nothing to do with psilocybes that are so much cooler to photograph and everything else. So yeah, definitely understand that. But so
that's it. You know, when you go out in the woods, it's a good entry point though. I mean, if somebody's going to get interested in psilocybes, that's like the gateway mushroom to like all of mycology. You know? Like, I mean, that's it. You know, if you're 18, I mean, I started out the same way, man. I was in college. I wanted to get high, you know, but after a while you start to be like, what the hell are these spores? And like, what was all this stuff about spore prints and colors and cystidia and basidia and like detached gills? And you're like, what is all this?
I mean, I definitely recommend the books that you were talking about and the identification guides, the Audubon stuff, to any beginners, because like hours in the field and identification guides, and then using your other resources to help verify that you're correct in your identifications are like the biggest things. But like people want to learn how to forage, go out and identify wild mushrooms. And I always try to tell people that time in the field spent is probably the most valuable thing, because, you know, going out and actually seeing them and comparing them to your guide when you're literally standing in front of the mushroom, you know what I mean, in the forest. Like I love that type of stuff, but there's also so much information contained and so many different species and everything else like that it's just almost near impossible to memorize it all to me. So I mean, I still to this day have to refer back.
There's no way you'll ever memorize it all. Anybody who thinks they're gonna like memorize all the mushrooms, even in their local woods, is sadly mistaken.
Yeah, for sure. I find things all the time that it's the first time I found them. You know what I mean? I still feel like what's exciting about going out and foraging, because, you know, you find new things all the time if you just keep going out and putting in the hours. Like not new things not discovered, but new things to you that you've never seen, you've never photographed, whatever the case may be. But,
I've hunted mushrooms with some of the world's leading mycologists, like literally like the top 100 myco people in the world, academic people who are specialists in one genus. And you find a mushroom and they're like, I don't know, it's something, you know, that AFF affinis or CF, like it's something close to that, or it's you know, in that group. But like people, that's why people go to school. When you go to get like a PhD in mycology, you study such a specific topic. It's not like you study, like even in the genus Lentinus that I studied, I only studied 3 species out of the 80 or so in that genus.
Wow.
because it was impossible. You're human, you can't, there's only 24 hours in a day. You can't know, even in that one genus, in those 3 species. Like, that was a 4-year PhD. I spent every day of that 4 years thinking about those 3 species of mushrooms. Like, I think people, you can get as deep as you want. Like, you can really, really get as deep. If you just want to forage and find stuff to eat, that's fine. But you can go this rabbit hole. Mycology. I just found a book yesterday that I didn't even realize was online, and I posted it. And my next live, I'm going to talk about it. And I finally figured out how to share the screen, because it's just, there's so much there that you wouldn't even, like, even me being a trained scientist, like, they were talking about like mitochondrial repair and these double-stranded breaks and enzyme, blah, 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 reactive oxygen species and all this. And I was like, oh my gosh, like, this is why my little culture sector. And this is why, like, when I go from T1, T2, T3, like, the morphology of my cultures changes. And this type of stuff, it's like, we in the Sonic Cube community, we touch on these things very superficially. But once you get into the literature, that's why I'm always talking about this literature. I have links in my YouTube videos. It's like, if you want to really get into this stuff, like, you will never, it's a hobby that you will never find like an endpoint. You'll never be like, okay, mycology, done with that. No, I know everything I need to know. That will never happen, ever.
No.
You could have 10 more lives and it would still never happen.
Yeah, for sure.
It's the Dunning-Kruger thing, right? You feel like you start to realize, like, oh my God, I don't know shit. Like, the more you learn, you're just like, I don't know fuck of anything. Like, I thought I knew what was going on, and I don't know anything.
Yes, it's funny. I don't know if you've seen that thing that was like, it's like a graph, and it shows the learning curve of mycology. Do you know what I'm talking about?
Yeah.
It's like
the Valley of Despair.
Yeah, you're at the very beginning. I'm really terrible at this. I'm really good at this. I'm great at this. Fall back to the bottom, and you're like, I know nothing, because you just have your mind exploded all the time by the amount of information that's out there. It's just once you have your head around one thing, you start to find that you really didn't know anything at all, because there's like 55 more chapters you've never explored. You know what I mean?
Oh yeah.
But
I usually have about 20 windows opened up on my browser, just like, well, I might get to read that. And then usually by the time my computer crashes, I'm just like, no, I'm not going to open up any of the, like, it's like restore Windows. I'm like, no, don't do that.
Not going back down that.
Just let it go, man. Like, I don't need to know about, like, oxidative stress and like phenotypic expression or whatever. Maybe next week.
Yeah. All right. So let's jump into some educational material for our viewers. Today we wanted to just like tip of the iceberg, talk about some really large subjects relatively quickly, but that we thought might be helpful to people who are trying to get into selection and breeding and crosswork. So I kind of wanted to make it so that there's like some context to the question. So we're going to imagine myself as me, somebody who has experience growing at home. I've done tons of work with different cubes and stuff like that, but I really want to get into isolation work and breeding, crosswork. So I'm going to ask Dr. Ed some questions. I have them written down, so I'm going to be looking down at the paper and asking some questions and seeing what Mr. Dr. Ed has to say about them. All right.
I've warned him, you guys. I could take like a 2-line question and turn it into like a 30-minute rant. So I'm going to try to keep it brief. So if some of my answers seem truncated, like, that's why. We only got an hour.
Yes, for sure. All right. So when identifying variants that you want to personally work with in your lab, what type of simple traits do you personally go for when you're making that decision about the cross work you want to do or isolation work you want to do?
The simple answer is I don't. I think after about 6 months, 6 to 8 months of doing mon crosses, I thought it would be more predictable, but it's not. Once you get to you do your mon cross and then you get spores, because those spores are a product of meiosis, you've reassorted all the genes in such a random pattern. This again is a very deep topic, but with a little bit of understanding of Mendelian genetics, the OPs like, F1 crossing, heterogeneous, big T, little t, that kind of thing. There's like no, probably back in high school, at least, people remember those Punnett square things, the little square thing, here's your gametes from one parent and here's your gametes from your other parent. When those things recombine with one single trait, it's very simple. Like say you have a Mendelian characteristics like height, you got a tall plant or a short plant. That's really easy to figure out. That's basic statistics. It's a no-brainer. But then when you start throwing in what they probably also taught everybody in high school, maybe in the last chapter of that bio book, a dihybrid cross. So instead of one trait, you have two traits so maybe you're tall and short and you're red and white or something like that. Now, when you take what we would think as like a simple trait, like potency or yield or resistance to contamination, those are not simple traits. So those are what we call polygenic traits. And without using a bunch of fancy words, they're not only the result of multiple genes, they're the result of also environmental factors oftentimes. They're the result of what we call epigenetic factors. So temperature and moisture levels might also affect your phenotypic expression, what the mushroom looks like. So you start off with these genetic factors. It's like nature and nurture, right? So you've got the genetics, which comes from your cross. That's kind of like the raw material of what you do in the lab or whatever. And then we actually grow the mushroom. How it gets expressed is sometimes very confusing.
Yeah.
It's not as simple. It's not as simple as we think it might be. Now, if you take two things, like if you take a GT and a B plus and you mate them together, you're probably going to get a brown cube, right? But if I take like an albino Louis Vuitton and maybe, I don't know, like a Riptide, who knows what the hell you're going to get? These are not simple characteristics. And a lot of them are also, it's kind of like you were saying with the ovoids, like you see something, you see a snapshot of that thing at a particular part of its life cycle. And it's really, really hard to draw conclusions based on, to correlate those, like that phenotype with what is actually happening in the genotype.
Yeah.
So, you see those ovoids and you pick them and maybe it was dry for 2 days before you've seen them. Yes. They were in a different location, you see that phenotypic plasticity is the fancy word. The look of that mushroom is highly dependent on the environment. Right.
Yeah.
And so drawing direct correlations between the crosses you make and what phenotype is going to be expressed is such a complicated thing that we don't even understand it in humans. Yeah. Right. Like we barely understand in humans how a single mutation, we have what are called SNPs, single nucleotide polymorphism. Like cystic fibrosis. It's literally like a single chain and a change in a single letter of that DNA sequence. At least I think it is. I might be confusing it with something else. There's a bunch of them that are literally one letter, an A, and instead of a T, it's an A, or whatever. It's a G instead of an A. Yeah. These things are so important, but how do you look at a mushroom and be like, I want to breed mushrooms to have this particular trait when we don't even understand where those traits come from?
Yeah, I think
it's virtually impossible.
I think that for a lot of people who are designating the two types of mushrooms they want to cross in regards to cubes and stuff like that is it heavily dependent upon the liking of characteristics of one, the liking of the characteristics of another, and just making that natural assumption that they're going to make a blend of the two. You know what I mean? They're like, oh, well, these two would look cool
Yes.
together. I mean, but that doesn't really play out like that most times. You know what I mean?
Number one mistake, I hate to say it, I'm gonna hurt some people's feelings, but they don't understand what a gene is and what alleles are and what Mendelian genetics are. This is not like mixing paint. This is not like you take a white paint and red paint and you mix it together and get pink. It's not the way genetics works.
Yep.
We know that. This was Mendel and many, many other people since him. Their contributions to genetics is we know that is not how it works.
Yeah.
Right. You can look at the classic examples of hemophilia, or, these things that are sex-linked traits. We didn't understand these things and we still don't understand most of them. But this is why we do like genetic testing, to mothers and fathers for things like, we know what alleles are, potentially for Down syndrome or breast cancer, or Alzheimer's or whatever, they're figuring out, this week. The problem is we can't do that with mushrooms. We can do that, but it requires a lot of money and time and skill.
Yeah.
So just like I like the way this one feels because I ate it one Friday night. Like the other thing is that even the testing for the pure alkaloids is not really well done and very accessible. So the subjective nature of like, oh, well, like I like Golden Teacher because they make me feel mellow, but I want to get really, really effed up. So let me mix that with Ghost. Or something like that, but then it's like it gets more complicated, right? Because Golden Teacher and Ghosts are little tiny white mushrooms. Like these kind of things, like people need to understand in the community that, and I'm sorry if this, but there's a lot more going on than just simply like, it's not like mix and paint. It just doesn't work that way.
Yeah. And that's just the thing is I feel like in the industry, people have tried to essentially simplify that method that is hyper complex, and there's a new cross coming out every day, and the work isn't there behind it. It's just I did this, here's one box, it's a new mushroom, and that's just that's not reality in regards to isolation work.
Yeah, I'm trying not to hurt anybody's feelings. I've been known to hurt people's feelings. I don't want to hurt anybody's feelings, but yes, there's a lot of work required. If you're going to call yourself a breeder, I was just on social media and I don't, what's the thing is, I have a PhD in mycology. I don't consider myself a mycologist. I don't get paid to do, indirectly, I don't get paid to do mycology. Like, I don't even consider myself a breeder. So when I see people writing on their posts, like, I'm a breeder, and I'm like, really? It's like the pollen chuckers in the cannabis world. It's like, what? You took a sandwich bag, put it on a male, or you just literally took like a male plant and you tapped it on a female. Like, I don't know if you really call yourself a cannabis breeder when you do that, like. So, I don't know if you do a double swab and you do now, nothing against double swabbing, ghetto swabbing, you take single swab and you swab 2 mushrooms. There's nothing wrong with that. But when you do that and then you grow what comes from that swab and you want to claim that you've created some new cultigen and you want to give it a name, like that's not really valid. There's a lot of steps to that. And there's a lot of ways that the community is going to come after you. But if you want to take that heat and you want to call yourself a breeder because you've double swabbed 2 types of fruit, then go for it. But people are going to not really accept that as a real the real method at a certain, unless you do. Now you can do a lot. Most of our cultigens have come from that method, double swabbing. There's nothing wrong with that. But if you're, say, crossing 2 tiny albinos and you're crossing them and you get another white little tiny albino, and you want to give it a name, it's probably not really valid, right? But, again, if that's what you want to do, You know, you get some dream, you got a name in a dream, and you're like, oh, I want to mix like stormtroopers with lucid gates and call it like Stormy Gates or whatever, you know, like
Yes,
go
exactly.
for it, man. I don't know. Name it after your hamster or something. I don't know.
Squibbles.
People do that, you know, people do that. Name it after your, like, you know, your wife or something. Don't do that. Or your girlfriend. Don't do that. We all know that. Don't get names tattooed on your body.
No.
I've got several friends that have their girlfriend's names tattooed on their bodies, and they regret it every day of their life.
No regrets.
See, I never. There's probably like 50 dudes out there like, yeah, fuck you, man. I got 3 girls' names tattooed on me. I'm sorry about that.
All right.
Don't drink so much.
Yeah, that's definitely a key takeaway for sure. All right. So before moving into crosses, let's say I want to create an isolation of a specific phenotype of a pre-existing variant. Where do you start?
I'm not really understand. What do you mean variant? I was trying to figure out, we discussed these questions before, and I don't really know what you mean by variant. Are you going after a particular phenotype or a certain characteristic or character?
Well, like,
I'm not really sure what you mean.
Well, like, let's say I have, I'm trying to essentially replicate a specific expression of a pre-existing variant, like type of mushroom. Like if you were trying to, like if you had a super interesting lucid gates and you just wanted to do, you know, that one of those, like essentially make like something where you were going after a characteristic that grew in a box that wasn't specific to that type of mushroom.
So why would you, if it's pre-existing, why would you try to make it again?
Well, I'm saying
that's
like
the part that's
something that grew extra thick and tall and had like a really thick stipe and all these other different characteristics that weren't typical of that mushroom, but you were trying to isolate those characteristics.
Clone it.
Yeah, just cloning. You had down.
Yeah.
You had spores, multi-spore, T0 plates, MS spawn, artificial selection, cloning.
Yeah. So my standard, and I think the standard, it doesn't matter if it's mushrooms or cannabis or orchids or tomatoes, the standard thing is to go basically through sexual. So I think you're talking about stabilization. Is that what you're talking about? You want to stabilize a certain phenotype, a certain look? Yes. How would you do that? The standard way to do that would be to clone. We're lucky, in fact, that mushrooms, because they grow, like essentially you have the sexual and asexual state in the same tub or whatever. So you can clone, let's say, the stem of the mushroom, and that will be literally a clone, an exact genotype. And we can grow that. You know, I've got plates and piles of that stuff sitting here. And so if you grew that clone in the same exact environmental conditions, then you would, it's like bananas, like Cavendish bananas, right? Every time you go to the store, you buy bananas, they're Cavendish bananas. You expect them to look the same, taste the same, like, you know, etc. So that's where cloning comes in. So that's where with mushrooms, we have the ability to do asexual cloning, just like they do with bananas and a lot of other and fruits and whatnot. So where you get into trouble is that, like, this goes back to the legal issues that you can't distribute live mushroom cultures in the US. So this has a historical context behind it, in that you would like to stabilize that fruit so that you can get spores from it. And so when I send you spores, you can regrow that particular phenotype. That's what plant breeding is. So if you go to the store and you buy, you know, I don't know, like a juicy, what's a tomato, like, let's just say like red Jubilee tomatoes. and you buy, say, like heirloom seeds, those heirloom seeds, because they've been inbred over and over and over and over and again, they get very what we call homogeneous or homogeneous. Their genes kind of become like a GT would be. Like a GT, you get GT spores, it's going to make GTs over and over and over and over again. Occasionally, you'll get a variant, a sport or whatever, you'll get some variation. But generally, if you take GT spores and you grow them out, you do a multi-spore plate, doesn't matter, multi-spore, you select a dikaryon, whatever, grow that, it's going to make little brown mushrooms, GT mushrooms. Now, if you're crossing things together, that becomes more complicated, because then you're taking an assortment of genes from, just like humans. I mean, probably a crude example, but if you took say two white people and you mated them together, you're probably going to get a white baby, right? But if you took a white person and an Asian, there's going to be a lot of variation there, because you're taking two distinct lines. So again, if you're made like a GT with a B plus, like they're pretty darn close, right? You're going to get another brown mushroom. But if you get further and further away from that kind of sort of common lineage, people have selected this way and they've selected this way, like with people, like Asians were selected by simple geography, right? So, those people have been separated, I mean, until recently for quite a while, and they've accumulated mutations and et cetera. And it's, very much more complicated, but you get the idea, right? Is that if you're trying to like cloning, what I think the simple answer is, if you want the same exact thing again, you clone. If you want to stabilize something, you clone something, and then you'll get spores from that particular mushroom. And then you go through the whole process again. And so you select, just like we would have for dogs, like you have a particular, let's say a multi-spore spawn run. So you'll get all your spores and you put them on a plate, and you've got a bunch of little dicaryons on there. And then you take that whole plate or part of that plate and you make what we would call multi-spore spawn. And then you grow that out and you'll see different phenotypes, and you find the one you like. So you would clone that, at least I would clone that as well as get spores from it. And then you essentially do the same thing over and over and over again. And every time you do that, you can go, most people, when they do the first cross or the first generation, they call it F1. And then when you go back to spores and you start again, you go up to F2 and then F3 and then F4. A lot of people used to say that by the time you get to about F8 or F9, depends what the organism is and how far, they were to begin with, that those traits could be stabilized. So at that point, you could send out spores and you could be like, look, here is my, my Tommy's Ranger mushroom or whatever. And it's going to, because it's been through all of these filial generation, and I've selected for the same phenotype over and over and over again, just like we have with dogs, right? Like it's the same idea. All these things are exactly the same. But if you let like a poodle and a Pomeranian mate with each other, like who knows what you're going to get? You surely are not going to get a poodle, and you're not going to get a Pomeranian. Yes. Right. But you might get something that looks kind of one way or somewhere in between. So you get this sort of spectrum of looks. And if you were a breeder and you wanted to make like a new kind of dog, you would take a particular dog and you would mate it with another particular dog. And you would do the same things. It doesn't matter if it's snakes or dogs or fish or mushrooms. That's where the work comes in. The work, like that process. Again, we're lucky with mushrooms in that whole spore to spore life cycle usually only takes about a month and a half or 2 months. Whereas with a dog, I don't know what the gestation period of dogs are, but it's like 6 months. And then you got to wait for that dog to mature sexually, another year or whatever. And so that process is slow. Like this is why we don't breed like elephants, because elephants aren't sexually mature till they're like 10 years old. So breeding elephants would be stupid. But we do have land races of elephants, right? Because they separated long times ago, a long time ago, and they've accumulated mutations, and they become, so you've got this homogeneous kind of look of an Asian elephant, and there's like an African elephant. But that's been happening for thousands or tens of thousands of maybe millions of years, right? We want to speed that up a little bit.
For sure. So just for, two follow-up questions. For more beginner listeners, right? So when I first got into the whole lab aspect of mushrooms and stuff like that, even just hearing the word cloning was kind of intimidating. I was like, well, there's there's no way I can do that at home. It's like that involves an advanced lab to do cloning. Like, you know what I mean? And like I didn't understand really what it was. It's just like you heard a word and you apply it to
Because
like
it has different contexts. In when we say cloning, we mean something different. There's molecular cloning, and there's cloning whole organism, and there's cloning the way we do it, and in plants. Somebody was talking about senescence earlier. Senescence in plants has a very different meaning than when we in mycology say senescence. We're talking about DNA degradation. With plants, they're talking about basically death. Like when a plant leaf senesces, that's a totally like, so cloning also has a very different meaning depending on the context.
Yeah, for sure.
So I would understand that in, what do they call it? Trepidation, that fear, because it sounds fancy, right?
Yeah.
It's not.
But for our listeners, tell them how simple cloning a mushroom is.
You literally just take a piece of the tissue. I did it like 4 times earlier today. You just basically, you take, let's see. So you're not gonna be able to see this very well, but I had a mushroom, and it's got like fog. And it's basically, I took a piece of tissue. That's, you just take a tissue. I took like a piece of the stem, and I just took a scissors that I flame sterilized with my dab torch, and I just like cut a piece of the stem off, and I put it onto agar, and then that started to grow out. And then I took a little that's not the camera, Ed. There's the camera. And then I took a little piece of that because it looks kind of bacterial there, which you would expect because it's a piece of tissue from a mushroom that was, grown in dirt. But yeah, you just basically take a small piece. It's like kind of like starfish, everybody knows like what starfish do, right? You can just like break off a piece and they like grow back. Like that's what our mushrooms will do. That would be cloning. But if you get to spores, so I think that's why I hate to say it, but a lot of people who are first beginning, they should really look in a basic biology textbook and understand the basidiomycete, the mushroom life cycle, not ascomycetes or zygomycetes, all that crap. Look specifically at the mushroom life cycle and understand what you're doing. The process of going from haploid and 2 haploids coming together, that would be like sperm and egg. They come together and then they form what we call a dikaryon. And what you would clone from a mushroom, the flesh inside the stem, that would be a dikaryon. So you can propagate that pretty much forever. It will get old, and assuming it doesn't get contaminated, you can keep that going pretty much ad infinitum, right? With some other techniques, water storage and whatnot. But if you go to spores, that's the little part of the mushroom life cycle you have to understand too. When you go from this thing that's called a dikaryon, which is basically mommy and daddy nuclei, they fuse together and then they become essentially a zygote. And then when that 2N or that diploid zygote, it becomes from dikaryon. This is another little different thing in fungi, at least basidiomycete mushroom fungi. Those 2 haploid nuclei, they fuse and they become like a little baby. And then those things separate kind of like a little baby. There's some steps that aren't exactly analogous to like humans, but anyway, it forms this 2N, this diploid nucleus, and then that undergoes meiosis. Meiosis, MEIOSIS, is where all the magic happens. That's where before, like in your dad's testes and in your mom's ovaries, like that is what happened to make these sperm and egg things. And so the mushroom does that on the gills. So in these specialized thing called basidia. And so you probably all see them. They got like little 4 prongs on them. I don't want to use all the technical words, but that process of meiosis, that karyogamy, which just basically means they fuse together and they undergo meiosis, that all occurs in this single one cell called the basidia or basidiome. Singular in the mushroom, and there are millions of those in a single mushroom. So in a single mushroom, maybe not millions, let's say 100,000 of those basidia. On every one of those basidia, you have 4 spores, and in every one of those 4 spores is a unique combination of new genetic traits. Now they could be very similar, right? Kind of back to the thing. If you made a GT, like a brown cube with a brown cube, you're probably going to get a brown cube. But if you mix, say, like a white person and a black person together, like you're going to get probably a lot of variation, and so when you often, like one of my old friends, she was mixed or whatever they're called now. And I don't know the politically correct word. Sorry, I haven't lived in America for 20 years. I don't know. She was half black and half Mexican, and she had a twin sister that looked very different from her, and her brother. She had two brothers. They both looked dramatically different. So when you take those two parents, and it was kind of a joke back then, because they were always, people were like, oh, that's not your real brother, and like, that's not your real sister, like that's what happens with mushrooms too. So occasionally you'll mix two brown mushrooms together and you'll get something like a tat, like a true albino teacher, right? You grow a GT for enough tubs and you produce enough brown mushrooms and enough multi-spore runs like they used to do. Because remember, only spores were the spores were the only legal thing you could deal with 20 years ago. So if you got hundreds and thousands of 18-year-old kids running these multi-spore runs and doing BRF cake, tech cakes and things like that with multi-spore syringes, inevitably something white's going to pop out. And that's where the tat comes from, the true albino teacher. And if you know anything about cubes, you know that like we have hundreds probably now of different cultigens that come from literally, maybe not, but figuratively, this single mushroom.
Yeah.
A single mushroom, right? Sure. That's where the whole tat lineage, so all of these things, and then there's various stories, and it's become like more like folklore now. But, the whole tat lineage comes from theoretically a single mushroom.
Yeah.
A single white GT. So when people start to talk about sometimes like what you would call a domestication bottleneck or diluting the gene pool, this I have some issues with, and I don't know if we want to talk about that, but it goes back to, I hate to say it, but a fundamental misunderstanding of genetics and the way things work. But I don't know if we want to go there. We're sorry. Dude, normally I'm on the live talking to myself, so I can go on for like hours without.
No, you're fine.
Nobody stops me.
So nobody stops. So
yeah, this is a great thing about doing your own live shows. Like nobody can stop you. You just talk for hours.
So you had mentioned the whole usage of F and familial generations and stuff. Can you walk people through that a little bit, and in regards to doing isolation work, what that means to move something through a familial generation?
So anytime you go back to spores, you can increase the F number. So the F filial literally means, it's the like Greek word for siblings. So you can think about it. It's kind of like a mommy and daddy who had a bunch of babies. So when, again, back to the single mushroom, like a single mushroom, which is derived from two parents. So you literally have two original spores that started out as haploids. They come together and they form that one mushroom. This is another misconception. That mushroom literally comes from two parents. But when you take those two parents and you mash them together to make that diploid 2N nucleus, and it undergoes meiosis, this is where we get all the diversity from. So every time you do that, and if you pick the same phenotype, so it's a little bit, I think, again, people kind of misuse, or they don't understand if you're going to say something like, oh, this is on the F6 generation, that means you went spore to spore to spore to spore 6 times, however you want to count it, many times. But what it means is you have to have picked for the same look, the same phenotype, right? So if you do a cross and you see that in your next generation, you see a phenotype that maybe you like better, well, you're essentially going back to F1. You can't really say, like, if you get to F4 and all of a sudden, like, your phenotype, your selection criteria changes, or you're, you know, something just doesn't start looking the way it's supposed to, or what you thought it was supposed to, or what you're selecting for, I mean, the F numbers become kind of irrelevant.
Gotcha.
Like, so just because something is on F8 doesn't mean it's stable, because stable for what?
Yeah.
Like,
Gotcha.
what is it stable for?
Yeah.
That's the problem is.
So when you're doing stabilization work, in regards to moving spores to the next generation, you need to ensure that you're essentially swabbing out fruits with the same phenotype over and over and over again, if that's what you're trying to
Yeah.
isolate for the rest of the look. Okay, gotcha. So
Yeah. If you're going for one particular look, you have to keep that look. You have to select, so you're going to select in every generation for that look. That's exactly what they do with plants, you know, like tomatoes or dogs or frogs or whatever. You have to keep the same criteria, and I think a lot of people don't they don't know what their criteria is, which is great, because maybe you get something you don't expect.
Yeah.
So you kind of have to have a very like defined thing if you are going to do breeding with a desired outcome, you know, like with a particular direction in mind. You have to stay with that. Otherwise, you're it doesn't matter. You don't have to stay with that. But if that's your goal, see, I don't have that goal. That is not my goal. I don't really. I in fact want diversity. Like, I'm looking for diversity. I don't want to stabilize anything. I think it's I'm not a I don't need 100 pounds next week. Some people are in that position, like wheat farmers, corn farmers. You know, they need a certain amount of product at a certain time. I'm not in that position, and I would never want to be. A lot of the commercial, like oyster mushroom growers and whatnot, they need to produce so many pounds a week. I'm not in that position, and if I was, I would stop doing mycology, because that's when you become a real farmer. Like, you could farm pigs or corn or wheat or rice, and that's not the job. That's not what I want to do. That's not what makes me happy. But if that's what people are looking for because they need to produce a product, then I don't know, more power to them if that's what makes them happy in life. But that doesn't make me very happy.
So you just go for interesting expressions. That's like what pretty much the most variable.
I don't even I don't even have a dude. Like, literally, I get monocaryon. I do my diamonds or my mon crosses. I really don't really. It's like I've got a lot of stuff going on, so it's kind of like I just look and I'm like, oh, I got my Thai monos. So I'll just do some diamond crosses with those, but I don't really like whatever I'm going to make spawn. Like, I might do a diamond with a true albino Melmac, or maybe, I don't know, a Riptide, or like, I don't really think about it much.
Gotcha. So it's more typically just like, what are you working on at that time? That's like invisibility, and then you're just like, yeah, let's do that. I like it.
Literally, it's like I just did a grab and drag to get like Thrack. I tried to get mono. I keep thinking that's the camera. Like the grab and drag to get monocaryons. Yeah, I just thought, like, yeah, I don't even know where Thrack comes from. Like, I ask on social media where it comes from. I don't know. So, like, it looks like a little portobello. It's like, that might be interesting. Try that.
I thought I saw one of the plates you held up at Stero.
Yes.
Nice. That's a good one to have a clone culture of. So now let me ask you this. Sorry, go ahead.
Oh no, I was having trouble. So that's another thing. I was having trouble with that culture. I had a little mite issue, and I've been finding it more. And so I cloned that because I had a tub of it fruiting. So one way to get back to make sure I have a nice, good culture, a good fruiting culture, I just cloned one of the mushrooms because I want to make that again. So yeah, that's what the C1, there's different, so people label things differently. So that's a clone of my Astero that was literally just fruiting. So I know that's another problem, is not all spore combinations will be the best combinations. But so that's where the diversity comes in. Like you might, like on your second or third flush, sometimes you'll get little things that pop up that wouldn't have popped up in the first flush. There's a whole other like 3-hour discussion.
I mean, agreed. And then in a lot of cases, you know, those second flushes, you know, renownedly will produce larger fruits. So like less filling the box, but larger fruits. I don't know if you ever heard that or not, but
oh yeah, that's all the time.
So you can definitely get some cool expressions out of getting multiple flushes out of a cake. And then it's funny that we were talking about this, but brings me back to something that I don't ever hear mentioned in the community anymore at all, which is probably something that you could talk forever about, but is the bioefficiency and like trying to build enough nutrient base in the substrate to like make it through multiple flushes, or if you're just trying to do that singular flush, how depth of cake and all that other stuff. But yeah, I don't ever, it feels like years ago, people were talking about bioefficiency all the time, and it just isn't really a concentration point anymore.
The gourmet people do a lot because again, they're back to really thin margins, you know, like if they can get 10% more out of their block, that's free money or lost money. I just heard some stuff earlier. Yeah. You know, trying to supplement your substrate, if that's what, like ours is not really substrate. Like usually with cubes, they're not actually eating the stuff. So it's more of a moisture reservoir. But yeah, if you've got 1,000 blocks and if you can pull like another ounce off each one of those blocks, like that's free money, you know, like if all you had to do is maybe change your pH or add some wheat bran or throw a handful of poo in there or something like that. But that gets tricky.
Yeah, for sure.
Like I'm not in it for, there's only so many hours in the day and so many years in your life. I got my hands full. Like I could do 3 to 4 times the amount of work, but you'll also burn yourself out.
Yeah.
Like new people don't realize how tedious things like washing tubs and like making substrate can get. When you have a goal in mind, it's great, but sometimes I feel people don't understand the amount of work that you actually have to do to do some of these things, and the amount of that work that is a little bit tedious.
Yeah. It's not all.
There's different ways around it, but depends what your outcome is. If you want yield, then wait. Like you're basically going to be a farmer. Like you might as well, you could be farming carrots or onions or chickens. That's what you're after. You're looking for weight. You're going to be you're a farmer.
Yeah.
So that's what people want. That's one way to go about it. I am so like the opposite direction of that. Like I don't care if I get 1 or 2 fruit off a tub. I'm happy. If that fruit makes spores, that means I can look for the diversity in the next generation.
Yeah.
If I've completed that life cycle and I've combined those 2 parents in a new genetic combination, like that gets me really excited. Like you can't imagine how excited, like, you know, a 50-year-old guy gets excited over a cotton bud, you know, a Q-tip, you know, a dirty Q-tip. It's like, you know, it's like, this makes me really excited. You know, like today I crossed like a Thai elephant dung with a true albino Melmac. And I found this swab, you know, oh God, I keep thinking that's the camera. That swab, that made me really, really, really excited. Yeah. Like, so, you know, if your partner or whatever sees dirty swabs laying around in plastic bags, please tell them, like, don't throw them away. Like I found that at the bottom of one of my old boxes, and somebody got a blob from this cross. So I made this cross and I started distributing it. And somebody told me they got a blob off it. So I'm like, let me try to find that. And yeah, so
nice.
Let those things out there, let them go feral, you know, and like people, that's kind of the reason I'm into the like breeding, if you want to call that. I love giving people the opportunity to find something cool, because like you were saying, most people are familiar with the basics and cloning and stuff like that. they might not have a microscope. They might not understand what the hell a haploid is or what a monokaryon is. So me, when I'm able to do this and I can mix things together and send them out to people. Like, I love that because I can't do it all myself. Like, there's no way you can physically do this. And that's when you realize, like, wow, I don't want all this, this cool stuff to go to waste. And you're like, let's send these out. And other people will have that opportunity, to look through that diversity that's there. And maybe lots of people, since I've been doing this like 2 years or so, people send me pictures and I'm like, wow, I never saw any, like, it's like, that was mine. And they send me these pictures of phenotypes and I'm like, wow, that's really, really, really cool. Like, I didn't ever see that because I didn't have the time to do it. But, you've got, like, you're a whole person and we're all different people, but I only got 2 hands, and 24 hours in the day.
Yeah, well, for sure. No, I did the Choose Your Own Destiny ghetto cross swab thing for the customers when I had a bunch of boxes going, like, hey, these are the variants I have growing right now. What would you like to see double swabbed? I'll send them out to you. You get to work them. I don't care about notoriety. You take it. If you can successfully get something out of the 2 of these, then it's yours. And actually, like, customers really liked it. They understood that there was nothing. It could either be one of the 2 or a mix of both, or be nothing at all. You know what I mean? But the ones who actually did grab them sent me pictures later of some pretty cool looking morphologies, and yeah, it was like, a couple of them did take. So it was cool to see them work with it, and then they were super proud of what they did, and I had never even grown them out. So I mean, it was like satisfying on their end. So, for sure.
I love that, dude. It's just like what we're doing, like what you're doing now at the podcast and what I do with my YouTube channel. It's just like getting the information out there so that people can have, I want to say, like, have a purpose in life, like something to do when you realize, like, wow, like, this is like something unique. Like, this is something that I should probably learn more about. And then you, as a byproduct of that, you learn a little bit about biology and maybe genetics and maybe ecology and maybe, you make a little money off it. Like, hell, I don't know. Give it a name. I don't know. Name it after your hamster or whatever the hell you want to do.
All right. So we're just rounding around an hour here. We got through like 2 of our questions, and it's okay because there was a lot of the information that was asked in the further questions was contained in your answers anyway. So you hit the nail on the head on a lot of them.
I was trying to roll them all into one there. Yeah.
No, you did great. You did great. So yeah, thanks for coming and talking today and hanging out with me and joining the SporeSwaps viewers and doing what you do. And we're always gonna be welcoming to Dr. Ed coming on the show and spilling his knowledge to the rest of us. So thank you so much for being on. And got anything else you want to add?
No, just if you guys want to like just my YouTube, I think I always forget. Oh, but you guys, I actually sell spores too. That's the thing. I'll do like these 6-hour live streams on my YouTube, and then I forget to ever mention that. Oh, by the way, like, I sell spores too. There's a whole bunch of crosses there and a bunch of like other people's cultigens that I've grown, and yeah, the standard kind of stuff. And yeah, you guys check out my YouTube too. I might go on there later tonight if I'm not too sleepy. But yeah, you guys check out my other my YouTube. It's just Edward Grand. That, for better or worse, it pops up straight away on the searches now.
So I'll put the link to your channel in the description for the YouTube posting.
Cool.
And so yeah, and also for anyone, if you just go to SporeSwaps.com and search that at the vendors, look for Dr. Ed's Spore Shop. He's got a lot of really cool stuff, a lot of hard work put into the items that are in his store. So check out his store. He's got a lot of good stuff.
Yeah.
But thanks, guys.
I might need to go pare it out a bit. It's getting out of control.
Yeah, me too. It's time for a purge. But thanks so
Yeah,
much
yeah.
for coming today, man.
Nice
Appreciate you.
talking to you. You
We'll hop
guys
on
later.
in. See ya.